something about the formation of blood cells, the process that's called as hemopoiesis or hematopoiesis, in which the word hemo or hemato stands for blood and poiesis stands for making or synthesis. So synthesis or making of blood cells is called as hemopoiesis or hematopoiesis. The process ordinarily starts in the bone marrow. That is, a newborn or an infant has a number of tissues in the body where the hemopoiesis can take place. These tissues are mainly the bone marrow tissues in the long bones like humerus or radius or tibia. This kind of bones, they majorly contain the bone marrow which can participate in hemopoiesis or hematopoiesis. But with age, this tissue goes on decreasing, and then it remains only in major parts of the axial tissues and the girdles, the pelvic girdle or some part of axial skeleton, that is some part of the thoracic cage, the sternum bone basically, they contain this hemopoietic tissue, hemopoietic bone marrow. Now bone marrow, what I see, is actually a tissue that's a combination of different types of cells. There are certain connective tissue fibers, there are certain cells, and the cells that are of this hemopoietic interest are the cells which are called as hemopoietic or hematopoietic stem cells. They are also called as hemo or hematocytoblasts. These are large cells with almost equally large or remarkably large nucleus. Now these cells, initially they are multipotent or totipotent you can call them. That is, they can get transformed into any type of blood cell, be it erythrocyte, be it leukocyte, be it thrombocyte, whatever cell can be derived from this stem cell. So these are multipotent stem cells which are initially present in the bone marrow. But depending upon the hormonal status, these can get transformed into something which is called as a committed cell. That is, its fate, its further development is defined. It is decided whether it's going to be a red blood cell or a white blood cell or a platelet. To start with, let's understand formation of a red blood cell, erythrocyte. So this cell that we're talking about as a committed cell is a cell committed to formation of a red blood cell. So now it becomes a myeloid stem cell committed to become a red blood cell. See, initially this cell is an irregular shaped cell with a very large nucleus in it. That is the hemocytoblast. Myeloid stem cell also looks similar. It then gets transformed into pro-erythroblast. Pro-erythroblast would be structurally almost similar, slightly maybe smaller in size, with almost similar nucleus. That is pro-erythroblast. This pro-erythroblast then gets transformed into a basophilic or early erythroblast. Early basophilic erythroblast. Now this early erythroblast structurally could be similar. A major change is occurring that we would come across in due course of time. This early erythroblast starts forming a large number of ribosomes in it. And these ribosomes start producing certain proteins which are essential for the process. So it's a cell that has accumulated large number of ribosomes and then gradually it transforms itself into late erythroblast. which now starts acquiring the hemoglobin. It starts collecting, accumulating large quantity of hemoglobin. Gradually, its color starts changing. Initially, it is bluish in color, or let us say a bit purplish in color. But as it starts accumulating more and more of hemoglobin, it starts becoming towards pink in color. So, a purplish or bluish colored cell starts gradually becoming pink in color. After accumulating adequate amount of hemoglobin, it becomes a normoblast. A normoblast. Structurally it will be similar. This is what a normoblast would look like. A normoblast, after its cytoplasm is occupied almost to 34% extent by hemoglobin, it becomes a cell that starts getting rid of its cell organelles, ribosomes, Golgi complex, most of the endoplasmic reticulum, and even nucleus. They are ejected by the cell. So it starts gradually transforming itself into an enucleated cell. It becomes a reticulocyte. So normoblast now becomes a reticulocyte. As it loses the cell organelles, the cell components, its central part gradually starts invaginating, gradually starts collapsing, and then it acquires its regular biconcave appearance. Now, up to formation of reticulocyte, it's a process that takes around 5 days. Reticulocyte to erythrocyte, a normal red blood cell, that process takes around 2 days. So within a span of about 7 days, the entire process is completed. Then the cells are released into blood circulation. In fact, even some of the reticulocytes are released into the blood flow. So blood may contain a few reticulocytes and a few matured red blood cells. The question is, how exactly is this process monitored? See, there is a trigger, there is a stimulus which activates the process of erythropoiesis or leukopoiesis or thrombopoiesis or whatever. That means in general the hematopoietic process it's triggered by some hormone, and that hormone in erythropoiesis happens to be erythropoietin. Erythropoietin is the name of the hormone. Now some amount of this hormone is contained in the blood. That is responsible for maintaining an appropriate level of RBCs in blood. Sustainable level of RBCs in blood will be maintained by erythropoietin contained in blood. Some amount of this hormone is generated in liver, but the major portion of this hormone is released by some cells in the kidneys. Now, how much of this hormone should be released by the renal cells, by the kidney cells, would depend upon variety of factors. One main factor would be the number of blood cells contained in blood, flowing through blood. In other words, the amount of hemoglobin they contain. In other words, the amount of oxygen they are carrying. See, oxygen conduction capacity of blood is entirely dependent upon the red blood cells. So the oxygen conduction capacity of blood cells or oxygen conduction capacity of blood would decide the amount of erythropoietin to be released by the kidneys. If the oxygen conduction capacity is declined, now how would kidney come to understand about it? If the kidney cells start facing the hypoxic state, if they start facing shortness of oxygen, then in response to that, they start releasing erythropoietin. It's abbreviated as EPO. This EPO would then trigger the process of erythropoiesis. The number of red blood cells can increase and the oxygen supply can be normalized. Erythropoietin level can be triggered, can be increased, can be enhanced by some other factors as well. Suppose if there is hemorrhage, loss of blood, bleeding, internal hemorrhage, whatever, and if the blood cell count is declining, then this can trigger more release of EPO, erythropoietin. If a person goes to a region which is at a very high altitude, where the oxygen content of the atmosphere is relatively lesser, the oxygen intake is proportionately less and oxygen conduction is accordingly reduced, then EPO can be released in higher levels. Aerobic exercise can trigger EPO release because that consumes quite an amount of oxygen. Oxygen level in the blood can decline and that would trigger the release of EPO further. So, release of EPO by kidney cells would depend upon a number of factors as I said. Normally, the level of blood cells, RBCs in blood would be appropriately maintained. Here we are talking about synthesis of blood cells. What about the destruction? Destruction is predominantly taking place in liver and spleen. Spleen is generally called as the graveyard of blood cells. A large number of blood cells are destroyed in the spleen. In liver also they are destroyed. And after the red blood cell is ruptured, its hemoglobin is separated. It's broken down into heme and globin. Globin is converted into bile pigment and it is suitably excreted, while heme would be stored in the liver and can be made available whenever fresh hemoglobin is being synthesized. So liver and spleen would be responsible for destruction of the red blood cells, especially the cells who have completed the span of about 100 to 120 days. While synthesis, as we saw, it takes a span of about say 1 week. Within 7 days, fresh red blood cells are synthesized. So that takes care of an appropriate balance in the number of cells contained in blood. Sometimes this balance can be hampered for a variety of reasons. A patient who is on dialysis, kidney cells are not functioning properly and the EPO release also would not be adequate. In such a case, the RBC number can start declining to an alarming level. In such cases, some artificially synthesized erythropoietins are recommended. It's called as a recombinant DNA synthesis of EPO. This can be injected into the patient and that enhances the erythropoiesis. This can take care of the disorder to some extent. Now, this artificially synthesized EPO, sometimes athletes also resort to this kind of treatment to increase the number of red blood cells in their blood, so that they can have more supply of oxygen, their performance can be enhanced to a better level. But then it has obviously a number of adverse effects. As more and more number of blood cells are synthesized as a response to the artificial EPO, they cause a kind of dehydration in blood. Dehydration in the sense major volume of blood is occupied by the blood cells, so water content is reduced. This dehydration can lead to accumulation of these excess red blood cells in certain places that may lead to blocking the blood flow. Ischemic condition can reach. That's a side advantage or that's a side effect of external EPO. Another thing is there's something called as blood doping. Blood doping means a person who is planning to go for some kind of an athletic event. He goes to take out some of his own red blood cells and keep them stored. New blood cells will be synthesized appropriately, and just before the actual athletic event, those stored red blood cells, he would get them injected into himself. So the number of red blood cells in his blood would be relatively larger. Oxygen conduction capacity of blood will be better, and the athlete would be able to perform better. This, of course, can be diagnostically confirmed, and it is found to be illegal. The athlete can be debarred from the event. If for any other reason the red blood cell number increases, that's a condition that's called as polycythemia. Polycythemia. Now, as we discussed, the hemoglobin accumulation starts at a very early stage. For this hemoglobin formation, two main components are essential to be present in blood. One is adequate volume of iron. Secondly, adequate vitamin B. Now, iron is in its isolated form, it can prove to be toxic. So it is stored in the body as a compound called ferritin or hemosiderin. It's stored as a molecule called ferritin or hemosiderin, and it is conveyed through blood in form of transferritin or transferrin, so that it can be suitably used whenever and wherever required. Whatever iron we consume through food, 65% of that goes for synthesis of hemoglobin. And that's how the appropriate rate of RBC production and destruction can be monitored. That's about hematopoiesis and destruction, that is hemolysis, balancing the relative number of red blood cells in blood. Do keep watching these videos, do like them, subscribe to them, and share them amongst your friends and family. Thank you.